AI again surprises us with another functionality. Can you guess what? From driving our cars to maintaining our homes, AI is everywhere. Now they will be in charge of laboratories too. Here is DeepMind's AlphaFold, which will fold proteins better than humans. We will know more about AlphaFold today. Google DeepMind, originally DeepMind Technologies, was formed in the United Kingdom in 2010 before being taken over by Google in 2014, becoming a completely owned subsidiary of Alphabet Inc. after Google's administrative transformation in 2015. In 2016, DeepMind applied artificial intelligence to protein folding too. Protein decoding is a centuries-old difficulty in molecular biology. We are talking about the newly invented AI technology AlphaFold. DeepMind's AlphaFold won the 13th Critical Assessment of Techniques for Protein Structure Prediction (CASP) in December 2018 by accurately predicting the structure of 25 of 43 proteins. This is a lighthouse project, our first major investment in terms of people and resources into a fundamental, very important real-world scientific problem, Chief Executive Officer of DeepMind Technology, Dimas Hassabis, said. The DeepMind team discovered that its previous approach, 2018, combined local physics with a pattern recognition-derived guide potential inclined to over-account for interactions among nearby residues in the sequence instead of interactions between residues farther apart along the chain. As a result, AlphaFold 1 selected models with somewhat more secondary structure, alpha helices and beta sheets, than was present. The AlphaFold 1 software design had several modules, each trained separately. They were used to generate the guiding potential, which was then coupled with a physics-based energy perspective. This was replaced by a set of subnetworks linked together into a single differentiable end-to-end model based entirely on pattern recognition and developed as a single integrated structure. In the 14th CASP in November 2020, AlphaFold's predictions earned an accuracy score comparable to lab approaches. The team outperformed all other groups in terms of accuracy. It received above 90 in CASP Global Distance Test (GDT), which measures the degree to which a computational program predicted the framework is similar to the lab experiment determined structure, with 100 being a perfect match within the cutoff range used for estimating GDT. One of the panel's scientific adjudicators, Dr. Andrei Kristofovich, called the feat truly remarkable and said the difficulty of predicting how proteins fold has been largely solved. The results of AlphaFold 2 at CASP were regarded as astounding and transformational. Proteins with fewer than 16 or more than 2,700 amino acid residues are not included in the database but are available in the entire batch file for men. Some researchers pointed out that the accuracy is insufficient for 1/3 of its predictions and that it does not provide the method of protein folding for protein folding problems. Still, AlphaFold has got many hearts, so artificial intelligence has got one more feather to adorn its hat. Two modules, thought to be based on an adapter design, are used in AlphaFold 2. These steadily refine a vector of data for each relationship between a residue of amino acids of the protein and another amino acid residue, and between each amino acid location and each different sequence in the input sequence alignment. Internally, these refinement transformations involve tiers that gather important input together and weeding out irrelevant data for these context-dependent correlations learned from training data. According to one article, the attention algorithm mimics the way a person might assemble a jigsaw puzzle. First, starting from small clumps like amino acids, then making it a larger whole. AlphaFold intended to add more sequences to the storage, initially covering almost all of the UniRef 90 set of more than 100 million proteins. 992,316 forecasts were available as of May 15, 2022. The open-source ROSE TTA-Fold and AlphaFold 2 software was released in July 2021, allowing scientists to run their own customized versions of the algorithms. DeepMind revealed a week afterward that AlphaFold had predicted nearly all human proteins along with the full proteomes of 20 other highly researched animals. That is quite an impressive achievement, and the structures have been made available on the AlphaFold Protein Structure Database. It was reported in July 2022 that the predictions of approximately 200 million proteins, encompassing nearly all known proteins, would be made available on the AlphaFold database. AlphaFold is still getting improvised. While the system currently has major constraints, the CASP results indicate that AlphaFold has the ability to assist us in comprehending protein structure and boost biological research right away. DeepMind intends to keep expanding the database with structures for newly found protein sequences and improve features and usefulness in response to user feedback. What is really this protein folding problem? Proteins are made up of chains of amino acids that dynamically fold together to form the protein's three-dimensional three-dimensional 3D structures. The 3D structure of the protein is critical to its function in biology. The protein folding problem refers to difficulty comprehending how the amino acid sequence determines the 3D structure. It entails comprehending the thermodynamics of the interatomic factors that define a folded stable structure. Through this very mechanism, a protein can quickly attain its final folded state, and the native structure of a protein can be anticipated from its amino acid sequence. Currently, protein structures are identified utilizing techniques such as X-ray crystallography, cryo-electron microscopy, and nuclear magnetic resonance, but these methods are both costly and tedious. Over the previous 60 years, such efforts have revealed the structures of approximately 170,000 proteins. In the actual world, there are more than 200 million known proteins. It will substantially aid scientific study if it becomes feasible to predict protein structure based solely on the amino acid sequence. However, Leventhal's paradox demonstrates that while a protein may fold in milliseconds, the amount of time it takes to calculate every potential structure arbitrarily to determine the true native structure is more than the age of the known universe. This makes protein structure prediction an urgent issue in biology. Researchers have used a variety of computational approaches to solve protein structure prediction over the years, but their reliability has not been comparable to experimental techniques, especially for small basic proteins. So eventually, all these methods got rejected. CASP, which was founded in 1994 to encourage the scientific community to generate their best protein structure predictions, discovered that for the most complex proteins, GDT scores of barely 40 out of 100 may be attained by 2016. AlphaFold began competing in the 2018 CASP with an innovative deep learning artificial intelligence (AI) approach. What if you cannot find the protein you're looking for? Instead of a specific uniprot accession, try searching by protein or gene name. If you don't find a sequence you're looking for, try utilizing the EBI protein similarity search tool against the AlphaFold DB sequences. If the requested sequence is already in AlphaFold DB, this may enable access to a structure prediction. If the query sequence is not accessible, a structured forecast for a sequence that is similar to the query may be offered. Check the complete proteome download for human proteins with more than 2,700 amino acids. Longer proteins are expected as overlapping fragments in this. Titan, for example, identified fragment structures as Q8WZ42-F1, residues 1 through 1400, Q8WZ42-F2, residues 201 through 1600. To know more about AlphaFold, we recommend you go through the Highly Accurate Protein Structure Prediction with AlphaFold paper published on July 15, 2021. We will talk about a few things mentioned in this paper. The AlphaFold architecture can be trained accurately using solely supervised learning on PDB data. By employing a technique akin to noisy student self-distillation 35, accuracy can be improved. A trained network predicts the structure of over 350,000 varied sequences from UniClust 3036 and creates a new dataset of projected structures limited to a high-confidence subset. The same architecture is then trained from the ground up using a mixture of PDB data and this new datasets of predicted elements as training data, with various training data upgrades such as cropping and MSA subsampling, making it difficult for the network to replicate the previously predicted structures. This self-distillation approach makes good use of the unlabeled sequence data and significantly enhances the final network's preciseness. AlphaFold used a blend of bioinformatics and physical approaches that mixes a physical and geometric inductive bias to construct components that gain insight from PDB data with minimal enactment of handcrafted features. For example, AlphaFold creates hydrogen bonds properly without a hydrogen bond score function. As a result, the network learns far more effectively from the minimal data in the PDB while also being able to deal with the varied and intricate nature of structural data. AlphaFold, for example, can handle missing physical context and build accurate models in difficult scenarios, such as entangled homomers that are proteins that only fold with the help of an unknown hem group. Because the PDB includes an array of constraints under which structures have been sorted out, the ability to handle unspecified structural conditions is critical for learning from PDB structures. Overall, AlphaFold is trained to generate the protein structure most likely found in a PDB structure. AlphaFold immediately outputs protein dimensions and makes predictions in the graphics processing unit (GPU) minutes to GPU hours according to the length of the protein string. This offers the intriguing prospect of predicting structures at the proteome and beyond. Isn't it exciting to know our science world is having another milestone covered? Comment down your thoughts. Also, do not forget to like this video, share it with your friends. Hope to see you soon. Bye!